My name is Dr. Justin Gainor. I'm the director of the Center for Thoracic Cancers at MGH, as well as the director of Targeted Immunotherapy. And today I'm going to be talking to you on frontline therapy for uncommon mutations. I'm just going to now share my screen. I just wanted to first begin with my disclosures. Beyond that, I think it's helpful. I think Dr. Sands provided us an excellent overview of the various genetic alterations, and I wanted to touch on a few key topics because they're going to have relevance for how do we go from having a mutation to then using a drug, and what actually happens, and what are some of the commonalities across these different genetic alterations, because even though these may be uncommon mutations that I'm going to focus on today, we can actually draw some common lessons. Okay, so the first thing is this central dogma of biology. This will be familiar to many of you, and this is just to say that we start with DNA. These are the blueprints. DNA gets turned into RNA, so from 2 strands to 1 strand, and then the RNA gets turned into protein. And so when we're talking about mutations, we're at the DNA level, and when we're using drugs like these quote kinase inhibitors or targeted therapies, we're blocking the resulting proteins. And so how might this work? Well, it's important to touch on how do normal cells first communicate signals to grow. So I have a schematic here where here's the surface of the cell, and we have a receptor here in purple, and essentially when something in its environment comes, it docks in, and then that creates this cascade. It's like a game of telephone where suddenly the cascade, and once it gets to the end, it says grow. Now, it's not as simple as this, because if you actually look at all of the various signaling in the cell, it gets very complicated very quickly, because it's not just this linear path. There are actually many different circuits. And back in the early 2000s, there was a lot of talk about how, given this complexity, we would never really be able to develop targeted therapies, because if you block one thing, if I block, up here near the top, then things can easily get around it, and nothing would really be able to, there were too many redundancies in the system. We've since learned, starting with the EGFR mutation that Dr. Sands described, that there's this thing called oncogene addiction. And what that means is so many of the genetic changes that we're describing, so again, here we have a schematic of the cell, and now here's a little blue receptor on the surface, and now we have a change, a mutation in that, where suddenly it no longer needs something to dock to it. It's constantly on. It's constantly sending those growth signals. And what ends up happening is the cell, the cancer cell, first, this leads to a cancer, and then secondly, the cancer becomes reliant on this pathway. It becomes addicted to it, and it becomes addicted to it because it's easier. There are no controls to it. There's no on-off switch. It's constantly on. So as a result, these cells preferentially prefer this pathway, and so then if we block it, the cells don't know what to do. So that's really the basis for all of the targeted therapies that you're hearing today, and that applies from EGFR all the way to some of these less common alterations like NTRK. The other thing I wanted to point out, and this is I think important when patients, families, providers alike are looking at their genetic reports, that these reports can get quite complicated, and that there are different types of mutations, and not every mutation essentially turns on that light switch. So I wanted to briefly talk to you a little bit about the different types of mutations, because the 4 genetic targeted therapies we're going to talk about today, involve different types of mutations, and sometimes the naming that we use for many of these gets confusing, and I thought just providing that background may be helpful. So the simplest type of genetic alterations is called a point mutation, and it's just that. Imagine, here's a gene, so a long segment of DNA, and there's just like one little change at one position. The analogy I give to my patients is imagine like a street, okay? And it's a very long street, and there are 600 houses on the street, and suddenly there's a change only at position house number 600. Right, so that's an example of a point mutation, okay? And the point mutations that are most meaningful is essentially taking the control switches and basically always turning that gene on. Another type of genetic change is called a rearrangement, and here the naming can get really tricky because rearrangements or translocations or fusions essentially all mean the same thing. Okay, so you may hear us say like an ALK rearrangement or an ALK fusion, ALK translocation, all mean the same thing. And what they're describing is here I have, say my gene of interest in green, and there's a second gene here, gene number 2. Essentially, what ends up happening is they get chopped up and recombined. So half of gene 1 and half of gene 2, and they come together. So it creates this fusion that's a mixture of 2 different genes, and that's just a different way of removing the controls on the gene. And then a third way that this oncogene addiction can occur is simply by amplification of the gene. So here, the gene is identical, so nothing has happened to that long street. It's just suddenly we have many more copies than we normally should. And that can also send increased growth signals to the rest of the body, to the tumor. This is a different version of the pie charts that Dr. Sand showed you, and so today we're going to be focusing on 4 different alterations. We're going to be talking about RET, we're going to be talking about NTRK, BRAF, and MET. So I'm going to start with the fusions just because the biology is similar, so I'm going to group those together and then we'll go to the others. So again, I'm going to start on the left here. So RET fusions, or RET rearrangements, these are found in 1 to 2% of lung cancers, but they're also found in other cancers. So they're found in thyroid cancer, and then they're found in many other, at very, very low frequencies, in many other cancers. We don't really see RET mutations, those point mutations, just those little changes in lung cancer. We see them in other cancers. But different genes can come together and be rearranged. So it's when you see things like this, KIF5B, CCDC6, it's just a fancy way of naming the 2 genes that have kind of fused together. Okay, so that's part of the nomenclature. What do we know about patients who have RET rearranged lung cancer? This is taken from a large series of over 160 patients. And what we know is that, in terms of age, that they tend to be about, a median age of 61. There's no gender preference between males and females, pretty even. They do occur more commonly in patients who have never smoked, and they are more commonly found in adenocarcinoma. This is just a schematic to show that, normally there are controls, and then in the setting of these fusions, it leads to cancer formation. Now, it's interesting, and this is kind of a historical note, but I think one that gives us some lessons. So back in 2011, it was December 2011. I distinctly remember it when RET fusions were first described, and at the time, the field of thoracic oncology already knew about ALK. We already knew about ROS1 fusions, and the initial papers that described RET seemed to relay a very similar biology. All three of those involve pieces of DNA that break apart and then combine together. And in the laboratory, it looked like they behaved very similarly. And so there was a rapid enthusiasm that, like, this is likely another target that we can use targeted therapies for. And there was a concerted effort to try to repurpose drugs. Basically, here's a whole list of drugs that were already approved for other things, but when we looked at the chemical structures, we found that they also targeted RET, and so it seemed like a great opportunity to, like, hey, let's just insert these new drugs. And while we saw a little activity, this is a waterfall plot. Some of you had probably seen these throughout the day, but essentially each bar represents a patient here. And if you're below 0, so in the minus side, this illustrates tumor shrinkage. If above that, that illustrates tumor growth. And in this first study of one of these repurposed drugs, it was a bit underwhelming. We saw only 28% of patients responding, and the responses were far too short. Not only that, is the drug was pretty toxic. And so, excuse me. And so, comparing this to ALK, ROS1, excuse me. Comparing this to other targets, it seemed like these weren't responding nearly as well. Excuse me. So there's a concerted effort to develop more targeted medicines against RET. So these are pictures that illustrate, these are all of the different genes that signal in that pathway that I showed you at the very beginning, that signal transduction. So these are trees, and each dot here represents the drug blocking one of those different kinases. And so what you see with these early RET drugs is that they actually blocked far too many things. Ideally, in your perfect targeted therapy, it blocks only one thing. And so that's what essentially was recently developed. So you can see these newer RET drugs, these were specifically designed to block RET. So pralsetinib and selpercatinib. And so you can see these same tree diagrams, but instead of blocking all these different things, they block very, very few. So these are and they really only block RET. So these are much more selective. And as a result, when you look at these in RET fusion positive lung cancer, now we get much different waterfall plots. So this is the kind of the same type of figure where each bar represents a patient, and you always want to see a waterfall plot where every single line is going down, and that's more or less what we're seeing here, is that the vast majority of patients are having very robust responses to these drugs. And this is just a similar waterfall plot, but looking at the activity within the brain, treating brain metastases. This is a different way of showing it, but essentially these are, each line means tumor shrinkage in the brain. And so both of these drugs, as Dr. Sands alluded to, both of these drugs were actually approved in the last 3 months to treat RET fusion positive lung cancer. And I would view them as now standards of care to treat anyone who has a RET fusion. So one can take a very similar approach. So for NTRK, like RET, NTRK is actually seen not just in lung cancer, it's actually seen in many, many different types of cancer types, as illustrated here, including some pediatric cancers. The thing about NTRK is, it's found very infrequently in some common tumor types. So it's very infrequent in lung cancer. But when you find it, it's incredibly important. So this shows you the frequency of NTRK fusion. So this is looking at over 4,000 patients tested, finding only 11. So the frequency is 0.23%. And these were all the different, fusions, all the naming the different genes that come together. So many, many different types. But, it's really important to still test for this, because as you'll see in the next few slides, when you find it, it can be transformative for the individual patient. These NTRK fusions are most commonly found in never smokers, patients pretty young, so 48, and with adenocarcinoma. But it also raises the question, if you have a genetic change that's so rare, 0.24%, how do you study that? Some of you may have heard of the different types of clinical trials. And this is what leads me to the concept of a basket study. And so the NTRK inhibitors were tested in a basket study, which is essentially you're testing one drug, usually, against one mutation, but in a variety of cancer types. So essentially, and I know you guys already had a talk this morning on tumor agnostic approaches, and this is really a tumor agnostic approach. So here's entrectinib. This is, you can hear it in its name, TRK. Here also the naming's a little complicated, but NTRK is the gene name, and that TRK is the protein name. So that's why you can see both listed there. So these are TRK inhibitors, and you can see in this pan-cancer analysis. So each bar representing a different patient, but you can see various different cancer types, showing very good responses across various solid tumors. So regardless of what type of tumor, it seems like patients were responding to the TRK inhibitor entrectinib, and in lung cancer, 70% had a response. The other TRK inhibitor is larotrectinib. It was the same type of design where various different tumors, actually 17 different tumors. Very nice waterfall plot. So both of those drugs are now approved, and I would argue it would be the standard of care for patients with NTRK fusions. The important thing is actually finding those genetic alterations. Now on to BRAF. So Dr. Sands mentioned that it's a specific type of BRAF mutation, and here we're no longer talking about those fusions. We're talking about a point mutation, so just one change, and that change occurs at position 600, and it's a simple switch from a V to an E, and then that sets off the cancer. So I bring up melanoma first because this is an important lesson compared to the NTRK experience and the RET experience that I showed you before. And this is actually one of those areas where speaks to the reason why we still have to do clinical trials. So here's one of the first trials of using a BRAF inhibitor in patients with BRAF V600E melanoma. So again, one of those waterfall plots, and we see everything going down. What about if we look at colon cancer? So, we saw close to 50% of patients with melanoma responding. In colon cancer, the same exact mutation, BRAF V600E, you use the same exact drug, but suddenly you're only seeing 5% of patients respond. So same exact mutation, but now it occurring in two different types of cancers, and we're seeing fundamentally different response rates. And so this is something that's also playing out with KRAS mutations now. It looks like KRAS mutations, these drugs, the newer drugs targeting them, have shown very promising response rates in lung cancer, but lower response rates in colon cancer, suggesting that there's some context dependence. So what about the experience in lung cancer? Is it going to be more like the experience in melanoma or more like the experience in colon cancer? And fortunately, what we saw in a series of studies shown here, it looks more akin to melanoma, seeing response rates around 40% if you just use a BRAF inhibitor, so just blocking that one genetic change. But through some very elegant preclinical studies in the lab, it was recognized. So normally, when the pathway goes, so BRAF, when this is active, it then activates MEK, and then ERK, and then GRO. So what if you not only block BRAF, but if you block MEK too? Well, in melanoma, what we saw was that if you use a BRAF inhibitor plus a MEK inhibitor, you actually did better than just the BRAF inhibitor. And so this has actually then been played out in lung cancer as well, another one of those waterfall plots. And what we're seeing, we're not comparing the two strategies in one study, but if you look in these single-arm studies where every patient got dabrafenib and trametinib, BRAF and a MEK inhibitor, much higher response rates. And so this is really the standard of care for BRAF V600E. Importantly, not every mutation leads to the activation. So in lung cancer, only half of the BRAF mutations are V600E, that specific change. The other half are not, and they generally don't respond to these drugs. So it's not enough just to see a mutation in a particular gene. It has to be the right mutation. And then in the last 3 minutes, I wanted to talk a little bit about MET alterations. And there are different types. So MET amplification is in about 1%, and then MET exon 14 skipping. And that already sounds a bit more complicated. So what are these? So MET amplification, I told you before, amplification just means extra copies of the gene. And so if you have extra copies, it can just send more signal. MET exon 14 skipping is a bit more complicated than that. I told you at the beginning that central dogma of DNA goes to RNA, goes to protein. Well, when you go from DNA to RNA, you actually lose some pieces, and those pieces, are called introns. And what ends up happening is, in that splicing process, when you're cutting them up, there can be mutations that lead to skipping one part called exon 14. And that's a really important part because usually it causes the cell to have to recycle MET. Every proteins in the cell also have some, get reused, they get recycled. And essentially what this means is you have extra MET on the cell surface, and so extra signaling going within the cell, and that kind of leads to tumor formation. So MET exon 14, there have been a series of MET inhibitors that have been explored here. Dr. Sands alluded to that crizotinib, wasn't designed as an ALK inhibitor. It was actually designed as a MET inhibitor. And so one of the first looks at that was looking at using crizotinib against MET exon 14. And here we actually see that again, this looks like an active therapeutic target that we can induce tumor responses. But the activity of crizotinib was not as robust as we had hoped, and so there are newer MET inhibitors, one called tepotinib. This was published in the New England Journal of Medicine. You can see here, and again, any waterfall plot where most of the bars are going down, that represents a promising drug. And so we can see here, tepotinib caused responses in about half patients. By responses, I mean below this 30% tumor shrinkage. It's arbitrary, but that's usually what the field has established. But that also means that all these other patients who are deemed not having a response, but are also benefiting because they're having tumor shrinkage. Geometry 1, this is a different MET inhibitor, but the same type of waterfall plots where most of these drugs, most of these bars are going down, showed about a response rate of 60% in patients who had previously not had any treatments. And so capmatinib was recently approved by the US FDA. It is now considered a standard of care for patients with newly diagnosed MET exon 14 skipping. Tepotinib has been given priority review status, but is not approved yet. So if we put all of the frontline FDA-approved drugs I'm listing here, and the preferred agents are in bold. For some, we don't know if one is better than the other, and so there I just list both. We're going to move to questions in a minute, but I just wanted to summarize that targeted therapies are now standard for patients with advanced lung cancer harboring RET, NTRK, MET, and BRAF. The success of NTRK and RET highlight tumor-agnostic approaches, but BRAF is a cautionary tale. As you heard from Dr. Sands, it's critical to genotype all these patients, and efforts are ongoing now to really understand kind of the next lines of treatment, such as resistance and overcoming resistance, and expanding this whole approach to other genetic alterations. So with that, I'll conclude, and I think we can open it up to questions.